Welcome to our lesson on protein synthesis in plants and animals. In this video, we'll explore the detailed steps involved in creating proteins. Protein synthesis occurs in two main stages, transcription and translation. Let's start with transcription, which takes place in the nucleus of the cell. During transcription, DNA serves as a template to create messenger RNA or mRNA. The DNA unwinds and unzips, exposing the gene that needs to be copied. RNA polymerase enzymes bind to the DNA and synthesize the mRNA strand. The mRNA strand is complementary to the DNA template strand. Once the mRNA is synthesized, it detaches from the DNA and exits the nucleus. Next, we move to translation, which occurs in the ribosomes. Ribosomes are made of ribosomal RNA, rRNA, and proteins. The mRNA travels through the ribosome, where it is read in codons. Each codon consists of 3 nucleotides and codes for a specific amino acid. Transfer RNA, tRNA, brings amino acids to the ribosome according to codons. Each tRNA has an anticodon that pairs with the corresponding mRNA codon. As tRNAs bring amino acids, they form a growing polypeptide chain. The ribosome catalyzes peptide bonds between adjacent amino acids. This process continues until a stop codon on the mRNA is reached. A stop codon signals the end of protein synthesis. No tRNA matches it. The completed polypeptide chain then detaches from the ribosome. It will fold into its functional 3-dimensional shape to become a protein. Both plants and animals synthesize proteins using these same basic steps. However, environmental factors can influence protein synthesis rates. In plants, light and nutrients play a critical role in this process. In animals, hormones and growth factors can speed up or slow down synthesis. Additionally, different types of cells have unique protein synthesis needs. For example, muscle cells require proteins for contraction and repair. Meanwhile, pancreatic cells produce enzymes vital for digestion. Gene regulation also impacts how much protein is produced in a cell. Enhancers and silencers are sequences that regulate gene expression levels. Transcription factors bind to these sequences, influencing mRNA production. In summary, protein synthesis is a complex but essential process in life. It enables organisms to grow, repair tissues, and carry out various functions. Understanding protein synthesis helps us appreciate cellular activities more deeply. Now let's go over some key vocabulary related to this topic. Transcription, the process of copying DNA into mRNA. Translation, the conversion of mRNA into a protein chain. Ribosomes, cellular structures where proteins are synthesized. Codon, a sequence of 3 nucleotides that codes for an amino acid. tRNA, transfer RNA that brings amino acids to ribosomes during synthesis. Polypeptide, a chain of amino acids forming part of a protein. Remember these terms as you continue your studies on biochemistry. Thank you for joining us today on this educational journey. Feel free to revisit any part of this video for better understanding. Don't forget to check our other videos on related topics. This knowledge is fundamental to biochemistry. Keep exploring and expanding your understanding of life sciences. Until next time, stay curious about the biochemical world. Goodbye and happy learning. End of video.